In this video, we will discuss about density-dependent specificity of GlyTR1 towards this beta-1 GlcNAc-branched and Glycan that are expressed on the cancer cells. In order to overexpress this beta-1 GlcNAc-branched and Glycan on cancer cell surface, they did different things. First one, they introduced this KIF uh, molecular small molecule that can produce high mannose glycan structures, thereby inhibiting this beta-1,6 GlcNAc branched and glycan, or reducing this beta-1,6 GlcNAc and branched and glycan. And in other things that they did is they knock down MCAT1. That will also it will also reduce this beta-1,3 beta-1,6 GlcNAc branched and glycan, and this TRAC CD3 knockdown and MCAT1 and TRAC CD3 knockdown will also leads to the reductions in the beta-1,6 GlcNAc branched and glycan. So first what it is, they try different combinations, and what they are able to find out that the optimized this GlyTR1 is shown to have a prominent or have the highest efficient functioning against the cancer cells. That is, and in this case examples they use L-PHA, which is a plant-based lectin, and they use the three different kind of linkers, L3, and finally they use this histidine at the end. And the specificity that they use is SCD3 single chain variable fragment. So they use this L-PHA that is bind to the anti-CD3, and this they termed it as GlyTR, and which is attached to the T cells. And once it interact with the cancer cells, it will release the cytokines to kill the cancer cells. First they check whether these things are generally expressed in the monomer forms, dimer form, or multimer forms, and they are able to find out that around 70% of this GlyTR1 are expressed in the dimer forms. And then they check this GlyTR using the Western blot and silver staining, and they are able to see that they are expressed majorly in the dimer forms. Then in order to check its specificity with beta-1,6 GlcNAc branched and glycan, they use three different conditions to reduce the beta-1,6 GlcNAc and branched glycan on the cancer cell surface or on the primary T cells. Then they use this MCAT1 knockdown, TRAC CD3 knockdown, and MCAT1 TRAC CD3 both knockdown. And compared to the control, they are able to see a decrease in the signal in all the three cases, indicating that the specificity of this beta-1,6 GlcNAc branched and glycan is density dependent. If the density is now reduced through the knockdown of any of the factors, its specificities or its specificities towards the cancer cells also reduced. And here they use another small molecule inhibitors to increase high mannose glycan structures, thereby reducing the density of beta-1,6 GlcNAc branched and glycan using KIF small molecules. And they can see that GlyTR binding without KIF is around 100%, and with KIF it reduced to 9%. And similar results they have seen for CD3 plus cells and CD3 minus cells. In both the cells they see the similar results, that is reductions in the specificity. And when they checked it in the Jurkat CD3 plus cells about these monomers and dimers, they are able to see that monomers binding efficiencies are lower compared to the dimers. So they continue with the dimers for the further experiments. Now they check the cell death percentage, and in this case the panel G they used MDA MB231F. It is a type of breast cancer cells, and what they are able to see that cells will increase for the both for the three donors that they have used compared to the only when there is no these GlyTR T cells are used. And when I did it for when I check the Glycan TR binding in this, uh, they are able to see that for the case of control, the Glycan TR1 binding is 100%, and in the case where they use a low amount of this CAR T cells, there is Glycan binding is slightly reduced. And for the other samples, here what I did is they started with the medium conditions. They can see some high amount of Glycan TR binding, but when they reduced it, they are able to see when they reduce the Glycan TR or CAR T cells with Glycan TR1, they are able to see less amount of binding. And same things they seen in its effect, and the similar effect they have seen in the dead cells. That is, the binding is high, the dead cells are high, the binding is low, dead cells are low. And for the second case also, it's the uh, binding is in the medium range, so dead cells are in the medium range. And for the B2, the binding is low, so the dead cells are low. The binding is low because the amount of GlyTR1 used in this experiment is low, and here it has shown the effector to target ratio is around 2:1 that they have used. Next, they wanted to see the activations of the CD8 plus cells in the presence and absence of these cancer cells. And what they are able to see is that when only the CD8 plus cells are used, the activated cells, that is CD25 plus cells, are very low, less than 25%. But when they are cultured with these cancer cells, and they can see the high amount of activations, that is CD8 25 plus activations is almost around 90% for all the three donors. So in the absence of cancer cells, the activations of these CD8 plus T cells are activations of this Jurkat T cells are low, that is CD25 plus uh, positive populations are low, but when they are cultured with the cancer cells, they are able to see the high percentage of CD25 plus cells. This indicating that the activations are also done in the presence of cancer cancer cells, and and this will also shows that their T cells can functions when and their modified T cells will functions more efficiently when they are activated or when they are cultured with the cancer cells. When they are not cultured with the cancer cells, they are not active and they will not be able to perform their functions of killing the cancer cells. So that's all for this video. If you need to understand more about the experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.